Welcome to our next lecture on RNA sequencing.
Please don't copy or distribute. So the learning objectives for today are to really understand why people sequence RNA, really to identify what are the different kinds of RNA that people typically target in an RNA sequencing experiment. More specifically, when we're looking at the expression of protein coding genes, oftentimes tools such as splice junction databases are used. So it's important to understand how they're constructed and how do we use them. More generally, I want you to get a broad understanding of the different ways in which people typically quantify relative expression of genes. And so again, when we're talking about using RNA sequencing to study, say, protein coding gene expression, there's a number of common but distinct ways that people use to take reads that you would generate from an RNA sequencing experiment and quantify how much does each gene get expressed in the cell at a given time. And finally, I want you to take away some understanding of what the effect of batch effects are and what exactly batch effects mean in terms of RNA sequencing experiments. So after we have a discussion about the general idea about bulk RNA sequencing, we'll then pivot to talk more about more recent technologies, more specifically in the field of single-cell RNA sequencing, which has kind of taken over the world since 2016, 2017. And so I want you to understand the general advantages of single-cell RNA-seq over classical bulk RNA-seq experiments, and also to gain some basic understanding of what the practical challenges of analyzing and collecting single-cell RNA-seq data is. More specifically, in terms of the classic single-cell RNA sequencing protocols, they can be broadly divided into two categories, one of which is called 3' tagging, and the other of which are called full-length transcript methods. So it's important to understand the broad difference between how these two types of methods work and what their relative advantages and disadvantages are. More specifically, especially when it comes to 3' tagging methods, which are typically higher throughput, a type of technical noise called dropout events are highly prevalent, and so it's important to understand what exactly a dropout event is, and in general, what are the sources of technical variation in gene expression measurements when you assay single-cell transcriptomes.
And so the first obvious question you should ask yourself is why bother sequencing RNA? And so the answer is that it's perhaps the easiest direct readout of the relative abundance of different transcripts in your genome. And so, relatively speaking, there are other kind of proteomics-based measures for measuring the abundance of actual proteins, but those are much more difficult to perform in practice, and they're much harder to generate comparable measurements across multiple samples or studies. And so RNA sequencing is a direct readout of relative abundance of transcripts, and people have figured out in large part how to compare RNA-seq data across multiple studies and things like this. Not only that, but sequencing RNA has a lot of different downstream applications. And so the most classic kind of experiment you would do after doing RNA-seq is you would do differential gene expression analysis. So differential gene expression analysis is the task of looking at gene expression measurements of samples from multiple groups. So this could be like a case control study, for example, and just identifying sets of genes that are significantly different in average gene expression between those groups. And so a couple of potential applications would be, for example, looking at the expression of genes before and after you treat cells with certain compounds to identify what the potential targets of those compounds are, looking at, say, disease versus normal samples to get an idea of how gene regulation changes with disease pathology, or looking at, for example, knockout mice where you've knocked out specific transcription factors and measured gene expression before and after to try to get an idea of what target genes might be regulated by a given transcription factor. There's a whole bunch of other applications beyond differential gene expression. So, for example, you can use RNA sequencing to identify novel transcripts, or even just identify regions of the genome that are transcribed that weren't previously known to be transcribed. And so, for example, in certain types of cancers, it's pretty common to see certain types of fusions between different genes. And so through sequencing RNA, you can identify these kind of novel fusions of different types of genes. You can also use RNA sequencing to identify different transcription start sites. It's also useful for observing RNA editing. And so what the central dogma doesn't really talk about is the fact that RNA isn't, even mRNA isn't necessarily just directly translated into proteins. Oftentimes there's mechanisms in place to edit RNA before it's translated. And RNA editing can have a wide variety of functions, including regulatory functions. RNA expressions oftentimes also used to detect what are called EQTLs, or expression quantitative trait loci. And so we touched on these briefly in the human genetics lecture, but basically a large part of the human genetics community is interested in characterizing the mechanism of action of how genetic variants associated with complex traits and diseases work. And so one way in which they do that is that not only do they genotype individuals and phenotype them at the organism level, but they also can use RNA sequencing, for example, to detect changes in gene expression that are associated with different genetic variants as a means of kind of identifying what potential genes might change in regulation with respect to genetic variants that sit in, for example, enhancers. RNA sequencing analyses are also used to study essentially time series analyses, where, for example, you might be interested in looking at how gene expression patterns change with continuous processes like, say, the cell cycle process or circadian clocks, or, say, differentiation of embryonic stem cells, or even reprogramming of, for example, iPSCs. And finally, it can be used in conjunction with some of the other technologies that we talked about in class. So, for example, you could use CRISPR-Cas9 to either introduce certain genetic variants or epigenetically shut down the activity of certain enhancers or promoters or so on. And then you could, for example, perform RNA sequencing before and after perturbation to understand what the effect of, for example, shutting down an enhancer is in order to identify what potential target genes there are for that specific enhancer. So here on this slide, I'll illustrate the basic RNA sequencing protocol. It's worth noting that RNA-seq has been going on for well over a decade now. And so there's actually many different RNA-seq protocols for many different scenarios, like if you're working with FFP samples versus just primary tissues. There's many different protocols for all of these different scenarios. But we'll just kind of talk about a vanilla RNA-seq protocol because a lot of the other protocols share a lot of similarities with this one. And so to get some of the terminology down, basically when I say transcriptome, I'm referring to basically all of the different RNAs that are being produced in the cell. And so this includes your classic kind of poly-A-tailed mRNA, but also includes a lot of non-coding RNAs that we'll talk a little bit about in a few slides. And so one of the most important distinctions here is that the transcriptome refers to all RNA products that are actually being expressed in a cell. And so this does not include all of the, for example, possible isoforms that might be produced from a gene with multiple introns and exons, but it really only focuses on the things that are actually being produced in a bunch of cells. And so the way that RNA-seq protocols typically start out is that you typically start out with isolation of total RNA, so that's basically any RNA being produced by the cell. And then you actually typically perform a selection step. And so the selection step is shown on the next slide. It's not shown here. But basically the idea is that most of the RNA in a given cell is made up of ribosomal RNA and RNA from mitochondrial genes. And so if your focus is, for example, poly-A-tailed mRNA, that actually occupies a very small proportion of the total RNA in the cell. And so if you want to just sequence poly-A-tailed mRNA, then you need to actually perform some selection for that. But once you've performed selection for your given target type of RNA,
Then you typically fragment your long transcripts into shorter fragments. And then what you do is because you can't sequence RNA directly, sequencing technologies are generally focused towards sequencing DNA. So you have to convert your RNA to DNA. And so you do so by basically annealing some random oligos to your RNA fragments, and then you extend them using reverse transcriptase. So that produces basically hybrid double-stranded molecules of both DNA and RNA. So at that point, you then add RNA-seq to get rid of the RNA, and then you add DNA pull 1 plus some primers to basically produce double-stranded DNA molecules, or double-stranded cDNA. And at that point, you then do some further size selection to select reads or fragments of a specific length. You then do sequencing, and then basically map those reads to the genome. And so because you're sequencing RNA and basically only a small fraction of the genome is typically transcribed, then most of your reads will fall to a small regions across the genome. And so we'll speak about this a little bit later, but you can either align reads back to the original genome if you don't know where the transcribed regions are, or you can map them to, for example, databases of just expressed regions of the genome, or even what we'll talk about later, what are called splice junction databases. But the idea is that, for example, if you map them back to the original genome, then in principle, the reads will typically align to, say, for example, exons of protein coding genes. And the idea of RNA-seq is that, roughly speaking, the number of reads that map to a given gene is roughly proportional to how often that gene was transcribed in the cells that you sequenced. And so that's not entirely true because, for example, genes that are longer in length, that have more base pairs, tend to have a higher number of reads, even if the number of transcripts is the same. But we'll get into that detail later. But the idea is that you can sequence reads, for example, map them back to the genome to figure out how often each transcript is expressed. And then in a differential gene expression test, for example, you could just compare the number of reads that map to a given gene across different samples from either cases and controls, and then test whether or not a given gene is significantly differently expressed in your cases versus your controls.
So when you perform RNA sequencing, oftentimes there's actually an RNA selection step. And the reason for this is that if you look at the amount of RNA produced for any given gene in the human genome, for example, you would actually notice that most RNA is actually attributed to a small number of genes. So for example, ribosomal RNAs or mitochondrial genes actually account for the vast majority of RNA in a cell. And so if you have no selection step, then essentially you're just going to be sequencing a bunch of ribosomal RNA and mitochondrial RNA. And so also, oftentimes when you actually try to extract RNA from a cell, you'll oftentimes have contaminant genomic DNA that you have to get rid of. And so on one hand, if you do a very light amount of selection, you can essentially try to sequence the total RNA within a cell. Or on the other hand, you could, for example, try to very specifically select for only RNA that have poly A tail, so for example, mRNAs. And so oftentimes, for example, certain selection steps are actually not even feasible. So poly A selection, for example, may not be feasible for degraded RNA samples. So for example, fixed RNA samples from, say, tumors, in which case you have to perform certain other selection steps like exome or cDNA capture.
And so again, when you do total RNA selection, that's basically just like not selecting at all. And again, in most species, ribosomal RNA makes up as much as like 95 to 98% of all the RNA molecules in a cell. And so for that reason, it's not that common for people to just sequence total RNA without doing some kind of selection or enrichment. The next kind of least selecting procedure is what's called ribosomal RNA reduction. And so here the idea is that you're trying to specifically deplete the ribosomal RNAs by hybridizing those ribosomal RNAs to specific oligonucleotides. And so the idea is that when you kind of capture these ribosomal RNAs and immobilize them, then basically all your other RNA molecules can be basically washed through and used as input to RNA-seq library construction. And so, again, depleting ribosomal RNA means that you're still left with a lot of RNAs that are going to map to, for example, mitochondrial genes, and you're also left with a lot of non-coding RNA that are not ribosomal, for example.
So in contrast to total RNA or the ribosomal RNA reduction strategies, poly selection is relatively specific. And so here the idea is that the vast majority of protein coding genes have poly A tails on their mRNA. And so you can select for poly A tailed mRNA specifically by using, for example, beads that have like poly T oligos hanging off of them. And so what will happen is that the poly A tails will anneal, they will hybridize to the poly T tails hanging off of these beads, and you can essentially select out these poly A tailed mRNA, and then only sequence those poly A tailed mRNA. And in doing so, you're essentially selecting just for that one species of RNA, and getting rid of both your ribosomal RNA as well as your non-coding RNA that are not poly A tailed, as well as most of the other non-coding RNA. So the final selection strategy is called cDNA capture. And the idea here is that you're capturing RNA from very specific targeted regions across the genome by, for example, designing arrays that have complementary probes to specific exons of protein coding genes, which will then capture RNA from those regions that you can then pull down and sequence. And so this strategy is used more often if you're trying to sequence genes, for example, that are very rarely expressed. And so if you didn't select for them, they account for such a small proportion of RNA in your cells that you basically never sequence them. And so another application of cDNA capture is basically when you have like a lot of heavy degradation of RNA from fixed samples. And so in those cases, this approach is basically more sensitive to picking up transcripts than general poly A RNA sequencing. But again, in general, cDNA capture is less frequently used, mainly because sequencing is getting more and more cheap, and our processing of even samples like fixed samples is getting better and better, and so it's typically not as necessary to have to capture and target specific transcripts unless there is one specific or small set of transcripts that you really need to quantify very accurately. And so bulk RNA assays have a lot of challenges associated with them. And so, for example, one of the biggest challenges associated with bulk RNA sequencing is essentially sample heterogeneity. And so here again, the idea is that in your bulk RNA assay, you have to collect RNA from like millions of cells in order to get enough RNA to do the sequencing. And so when you're working with primary tissues, for example, it's rare or impossible to find a tissue or a million cells that are all identical. And so typically there's multiple cell populations present. And so it's worth pointing out that this is true even when you do things like FACS sorting to sort out cells that express certain cell surface markers, because there is no such thing as there basically is no such thing as a set of cell markers that uniquely identify exactly one cell population and doesn't capture any other type of cell. Basically, you should assume that whenever you're doing bulk assays, there's always multiple distinct cell populations present, and the expression, the transcripts that you sequence are from a collection of distinct cell types. And so this is a problem because, for example, if you are trying to compare how gene expression varies across samples, then what you're actually measuring is both a potential change in gene expression within each cell population, as well as you could also be picking up just changes in proportions of these different cell populations. And so essentially, population heterogeneity or sample heterogeneity is essentially a confounding factor in gene expression analyses. Another potential problem you have is one that we talked about with ChIP-seq. So for example, you might have multi-mapping reads. So reads, especially if you're doing short read sequencing, might map to multiple regions in the genome. And this happens pretty frequently. And so because it happens so frequently, how you quantify transcript expression using these multi-map reads can basically, your quantifications can change significantly depending on how you deal with those multi-mapping reads. Also, alternative splicing turns out to be a pretty big problem because currently the vast majority of RNA sequencing experiments are still done with short read sequencing. And so alternative splicing is difficult because, especially if you have genes with a large number of exons, it can be hard to quantify, for example, individual isoform level expression because your reads are short. And so at most your reads might only span one junction. And so it's impossible to distinguish expression of isoforms from genes with many, many, many exons. Furthermore, it can, depending on whether those isoforms have already been characterized. So depending on whether someone's been able to distinguish which potential isoforms are not expressed versus those that are expressed, then you may not, may not be possible to even identify which splice junctions are actually being expressed, never mind entire isoforms. And so another classic problem of RNA sequencing is that you can have a huge range of gene expression patterns across the different genes in your cell. And so, for example, ribosomal genes and mitochondrial genes are typically some of the most highly expressed genes in any given individual cell. And so they tend to dominate the reads that you sequence in RNA sequencing experiments. So even when you deplete ribosomal RNA, for example, oftentimes you don't deplete mitochondrial genes, and so our mitochondrial genes essentially soak up most of the reads that you sequence. And so that essentially makes it harder to detect expression of more poorly expressed genes. Some other problems involve measuring expression of different types of RNA. And so historically, a lot of people have focused on expression of protein coding genes, but we now know that non-coding RNAs are actually more numerous than protein coding genes. And so it can be actually hard to capture small RNAs. And so typically you might have to perform multiple selection procedures in order to sequence, say, poly RNA versus small RNA, and so on and so on. And finally, again, as I mentioned with the RNA integrity slide, it's typically hard to get really high quality RNA across many samples. And so when you're comparing transcripts and gene expression patterns across multiple samples that you might collect, RNA integrity and diversity, library complexity essentially, can change between samples. And so this can affect how accurate your comparisons between samples are.
So I mentioned on the previous slides that one of the steps of the RNA-seq protocol is to map reads from an RNA-seq experiment to the genome or transcriptome in order to identify where the transcript came from. And so there's a few ways to approach this mapping task. So the first approach is basically just to avoid mapping at all and just do what's called de novo assembly. And so this is analogous to the whole genome assembly approach we talked about previously, where the idea is you take your shorter long reads from your RNA-seq experiment and you basically just try to rebuild the original transcripts that the reads came from. And so similar to the whole genome assembly problem, this approach essentially relies on very high coverage of all the transcripts that you're trying to rebuild. And so typically in practice, you can only really build the highly expressed genes because those are the transcripts for which you have enough coverage of your transcript in order to really identify your contigs and rebuild your original transcript. In contrast, most of the poorly and even moderately expressed genes don't have high enough coverage across the entire transcript in order to rebuild the entire transcript. And so basically, although the advantage of de novo assembly is that you don't need a reference genome to map reads to the transcripts, the primary disadvantage is that you can only really rebuild highly expressed genes from the reads. And even if you use long read sequencing, part of the problem is that, your long reads avoid a lot of the assembly problems that you would have with short reads. But typically you're able to sequence fewer transcripts when you use long read sequencing. And so you still may miss a lot of the transcripts that are poorly expressed. The second approach you can use is to basically just take reads and map them back to a reference genome. And so here you might just take each read and align them to the genome and hope that most of the reads align uniquely to the genome. And so the main challenge you have with mapping reads back to the genome is that reads that map entirely to, for example, one exon may be alignable, but the bigger problem are the reads that span splice junctions. Because the problem is that the alignments of splice junction reads back to the genome, the true alignments anyways, will involve large gaps, because for those reads that span splice junctions, part of the read will map to the 3' end of one exon, and the other part will map to the 5' end of another exon. And because exons are separated by potentially large introns, then those gaps are going to penalize the alignment very heavily. And therefore, in practice, it would be impossible to find those kind of gapped alignments, essentially. And so mapping back to the original genome essentially involves basically missing a lot of alignments of spliced reads, unless you do something about this. And so in practice, a third strategy is typically used, which is basically to map reads to a transcriptome database or a spliced junction database. And so the general idea is that for most genomes, many of the model organisms anyways, you typically know where most of the, for example, protein coding genes are. And so you typically also might know what the general intron and exon structure of most of those genes are. And so the idea is that either theoretically or using previously collected data, the idea is you take your genome sequence and you build possible isoforms out of those genes. And so the two ways of doing this are either theoretically or observed. And so the theoretic transcriptome databases basically just says, okay, well, if I know where all the exons are, then I can build all possible hypothetical transcripts that you could make by combining all of the exons in all the different ways that they can be combined. And then basically I'll just make a huge database of possible transcriptomes. And then now when I have a new RNA-seq experiment, I can take the reads, I can just map them against all of the possible isoforms. And I can be fairly certain that I'll cover most of the possible isoforms that are being generated. In contrast, the observed approach just says, okay, well, maybe in the particular tissue or cell type that I'm looking at, maybe someone has already done really deep RNA sequencing of those tissues or cell types. And so I'll just look at what transcripts other people have previously identified, and I'm just going to build a database out of isoforms that I know have been sequenced already. And so the advantage of the theoretic transcriptome-based approach is that you don't need to rely on someone having done deep RNA sequencing of your tissue or cell types before. Furthermore, even if someone has done RNA-seq experiments before on your sample of interest, they may not have been able to characterize all the different isoforms that were expressed. So if they were using like short read sequencing, it would be really hard to actually identify which isoforms your reads came from. The best you can do is pretty much just identify which splice junctions or exons an individual read map to. So the nice thing about theoretic-based transcriptomes is that you can be sure that you can build a pretty comprehensive database of all possible isoforms, and you don't need to rely on other people's data. The advantage of the observed transcriptomes is that you're transcriptome databases tend to be a lot smaller. And this is an advantage because if you have a read that, if you're building a database of theoretic transcriptomes, for example, there's going to be a lot of sharing of splice junctions or exons between different isoforms. And that's a problem because if you have a read that came from a single splice junction of, say, exons 1 and 2, in theory, there could be a lot of isoforms that have exons 1 and 2 spliced together. And so therefore, when you map your read to your theoretic transcriptome database, then you'll have a lot of multi-mapping reads because there's a lot of possible isoforms that have exon 1 and 2 spliced together. But if you only look at observed transcriptomes, for example, maybe previously people have found that exons 1 and 2 only get spliced together in one particular isoform, and all the other possible isoforms that could be made were not actually made. And so your observed data already tells you that you could ignore the other possibilities. And so basically, because the observed transcriptome database is much smaller, multi-mapping reads will probably be less of a problem. But the disadvantage of the observed transcriptome database is that you might miss novel splicing events that weren't previously seen in other data, right? And so basically, yeah, there's advantages and disadvantages to both the theoretic and observed approaches, and so it's both of these approaches are used in practice. The only thing I want to say about splice junction databases is that, like I just mentioned a few minutes ago, whether you use a theoretic or observed transcriptome database, if you are using short read sequencing where the reads are much shorter than the transcript length, then in practice it's pretty much impossible to quantify isoform level expression measurements because isoforms are very long, and so because the reads are so short with short read sequencing, it's generally speaking, there can be many isoforms that a given read maps to. And so multi-mapping basically prevents you from assigning expression to whole isoforms. So in that case, oftentimes people actually don't map to databases of whole isoforms, they map to databases of splice junctions, where different either theoretic or observed pairs or triplets of exons that get spliced together are used to map reads to. So one of the most frustrating aspects of working with RNA sequencing data is that RNA sequencing data inherently has a lot of variability, right? So no two RNA-seq experiments are going to give you the exact same result or exact same expression measurements. And this is basically due to both biological factors and technical factors. And so the point when you're doing an RNA-seq experiment, you intuitively want to get some kind of statistical confidence about each expression measurement that you make. And so in order to get that statistical confidence, you have to make every measurement multiple times and vary different factors, either technological or biological, so that you can get a sense of, how good is my estimate as to how highly or poorly expressed each one of these genes is, right? And so suppose that you're doing like a case control experiment where you're trying to compare normal versus tumor lung samples in humans. Right? So what you would do is, number one, when you're doing these RNA-seq assays for multiple samples, what you do is you sequence each sample like multiple times, and each time you kind of vary technological factors. And so, for example, what you could do is you could take like a normal lung sample, you could extract out total RNA, divide that total RNA into two different pools, and then have two different graduate students perform RNA-seq on each sample individually in order to then get an idea of how much variation is there between different graduate students measuring RNA-seq on basically technical replicate samples. And similarly, you could, for example, divide samples among multiple flow cells to determine how much variation there is between flow cells for the same sample. Similarly, you'd also vary biological factors. Like obviously you would collect samples that are both from normal lungs and tumor tumor samples from lungs in order to get an idea of how much variation is due to normal versus tumor. You might also collect samples from both males and females to see what kind of gender-specific differences, to see what the effect of gender-specific differences are on gene expression measurements. And so by looking at both the technical and biological replicates, a lot of RNA-seq software will be able to give you some statistical confidence about, how certain is the software about different expression measurements, and therefore they'll be able to give you confidence about, if you're measuring like a differential gene expression analysis between tumors and normal samples, how confident are you that the fold changes that you estimate are real? So one of the most frequent use cases of RNA-seq is to identify differentially expressed genes between two or more conditions. And so, for example, if you were interested in identifying the targets of a particular transcription factor, one way to do that would be to measure the expression level of every gene in the genome before and after you knock down or knock out that TF. And so suppose in this hypothetical example that you perform 500 RNA-seq assays on different samples within either the wild type or the mutant. And in this particular case, you're looking at one specific gene and you want to ask the question, is this gene differentially expressed between cases and controls? And so the red curve here represents the hypothetical measurement of that gene's expression after knocking down this particular TF. And the blue curve represents the expression level of that gene when in the wild type, so when you didn't knock down the TF. And so here I just want to illustrate the concepts of effect size and significance with respect to differential expression testing. And so here, the effect size could be roughly understood as the sort of the gap between the means of the blue curve versus the red curve. And so a little bit more formally speaking, you could measure effect size as essentially the log fold change here, or in this case will be the log ratio of the expression of a gene in the mutant versus wild type. So in this case, for example, that would come out to something like minus 1.6, which would tell you that there's a drop in the expression level of this target gene. On the other hand, the notion of statistical significance could be essentially visualized as the overlap between these two curves, the blue and the red curve. And so intuitively, the less overlap there is between the red and the blue curve, the more statistical significance you would get from that statistical test. And similarly, if there's more and more overlap between the red and the blue curves, then the statistical significance is less. So the p-value would be larger. So one of the trickiest aspects about RNA sequencing experiments is trying to decide how deeply do you need to sequence each individual sample. And so the take-home message of this slide is essentially that no matter what you're trying to measure, you should basically keep sequencing until the thing that you're trying to measure stops changing significantly, right? And so to give you an example, if you are interested in detecting alternative splicing events, right? And so your goal isn't so much about quantification of gene expression, but it's about identifying all the different splice variants that occur. Then one practical way to determine what your average sequencing depth should be is you could, for example, take a few samples out of your hopefully large collection of samples that you're going to sequence. And you could sequence them very deeply, right? And then what you could do is you could run a bunch of simulations where you take your reads that you were able to align against the genome and just start subsetting them. Start subsetting the total number of aligned reads down to start at say 5%, then go to 10%, 20%, 30%, 40%, and just calculate on average, if you only sequenced 10% of the reads you did or 20% or so on, how many, either known or novel splice junctions could you identify with that subset of samples? And basically, when you do this enough times, you should get a curve like you see on the right, which basically starts out steep because when you sequence very few reads, you should be detecting new things all the time. But at some point, you should start to notice that even if you increase your sequencing depth, you don't really discover anything new. And so these curves basically give you an idea of your trade-off between sequencing depth or cost and measuring the thing that you care about, which is on the y-axis. And so again, basically what your effective sequencing depth should be is basically where you think that sweet spot is, where you essentially don't get that much more by sequencing more. And so it's worth pointing out that this is a pretty different approach from DNA sequencing, for example, where in the case of like genome assembly, you could basically look at coverage of different regions of your genome as you increase sequencing coverage or you increase sequencing depth. And it's fairly intuitive when you need to stop sequencing. But for RNA sequencing, it really depends on whether you're, for example, trying to do quantification, in which case you'd be looking at the variability of your expression measurements as you increase the depth, or in the case of detecting splicing events, then you would draw curves like this that I'm showing you here.
So let's take a few minutes to talk about visualization of RNA sequencing data. And so when you perform RNA sequencing, you're essentially measuring the expression levels of thousands of genes in the human genome for every sample that you sequence. But when you actually draw visualizations of your samples, like in a paper, like these plots that I'm showing you here, in which each sample is a single point, you're typically looking at 2D scatter plots. And so how do you go from samples with thousands of measurements down to 2 measurements that fit onto a 2D scatter plot? So that task of visualizing high-dimensional data is known as dimensionality reduction, and PCA is one of the most common strategies that you'll hear about for doing that kind of visualization. And so for illustration's sake, suppose that we're looking at samples that are sequenced from a case control study, where, say, the blue points are cases and the red points are controls. And suppose that in this hypothetical genome, it has 2 genes. And so here, all of the samples then can fit on a 2D scatter plot without any kind of fancy visualization, but we're just using 2D scatter plots for illustration purposes. And so note along that each of the x and the y axis is a projection of all of the samples onto just that axis, which in this case means that for each one of these axis plots, what you're looking at is the distribution of samples along that gene's individual expression level. And so it's pretty clear that neither gene A nor gene B distinguish the cases and controls, but intuitively, when you look at the whole scatter plot, you can see that there's pretty clear differences in expression patterns of the cases and controls, right? And so here the cases have higher expression of gene B and lower expression of gene A compared to the healthy controls. And so PCA is essentially a technique for redefining new axes that kind of capture what we call the directions of maximum variance. And so in 2 dimensions, the axes PCA defines are called PC1 and PC2, or principal components 1 and 2. that are shown in text. They're labeled in red text on the figure, and also they're drawn as black lines on the plot. And so you can see how the long black line that's labeled PC1 is in some sense following the biggest direction along which points scatter in the plot. And so this is called the direction of maximum variance. And so PC2 is the second direction where you see a lot of scatter of the points in that direction in the plot. But PC2 and all other PCs are always kind of restricted to be orthogonal to all other PCs. And so what that means in 2D is that PC2 has to be at a right angle to PC1. So now on the right-hand side of this slide is a new scatter plot that's drawn with respect to our new axes, PC1 and PC2. And so if you take a step back and you look at these 2 plots, you can see that the PCA plot on the right is essentially a rotated version of the plot on the left. And so if you took the plot on the left and you rotate it something like 45 degrees counterclockwise, then you'd get the plot on the right. And so the take-home message here is that PCA is intuitively a rotation of your original plot or your data. And so the second thing to notice here is that if you look at the 1D scatter plot along the x-axis of the PCA plot on the bottom right, which is labeled as principal components 1, and you can see now on that one-dimensional plot that the cases and the controls actually separate perfectly along this dimension. And so secondly, the visualization of the data along only PC1 in which you see this separation of the cases and the controls, essentially captures the essence of the original scatter plot on the left, right? So we said earlier that this on the scatter plot on the left, you could kind of intuitively see that the cases and controls naturally separate, even if not just by gene A or gene B. And now you can see PC1 essentially does capture that separation of the samples, even though PC1 doesn't correspond to an individual gene A or gene B. And so, in summary, PC really, in this case, helped us redraw the 2D scatter plot on the left into a 1D scatter plot on the bottom right, while still kind of capturing the essential distribution of the cases and the controls. And so hopefully these two slides gave you some intuition for how we can visualize high-dimensional data in lower-dimensional scatter plots by essentially throwing away, for example, axes or directions of less variance. And so throughout this course, I've tried to make the point that regardless of which genomic assay you're looking at, in terms of the downstream analysis of the results of those assays, batch effects can play a huge role in terms of driving variation between samples. And if you don't address them, then they can cause significant changes in your conclusion of your study. And so here's an example of a fairly well-known paper now, where the authors of the study basically did RNA sequencing on a number of different cell types and tissues in human and mouse. The goal being to basically try to characterize whether expression in the same tissue across human and mouse is basically more similar than between two different tissues from the same organism. So either, mouse or human. Right? And so this PC plot from their paper basically illustrates or visualizes the different RNA-seq samples that were collected in different tissues for human and mouse. And so broadly speaking, the circles represent tissues from human and the triangles represent tissues from mouse. And you can broadly see that the circles kind of cluster together in this visualization and the triangles cluster together in this visualization. And so here they're showing the first three principal components of the expression data. And again, the main point is that the human tissue samples seem to cluster together and the mouse tissues seem to cluster together. And so the broad conclusion of this paper with respect to this figure was that gene expression patterns are more similar among different tissues within the same species than between the same tissues across the two species. And this paper was somewhat controversial in the sense that there are many papers before it that studied conservation of gene expression patterns in tissues across organisms, including humans and mouse and other vertebrates. And the broad conclusion that people had made up to that point is that gene expression patterns are pretty highly conserved. And so generally speaking, people thought that the heart expression of human looked more similar to the heart expression of mouse than to the, say, liver expression of humans. And so a few days after this paper was formally published, people started digging around the data a little bit more, and they figured out that a large part of the reason for this visualization, the reason why humans cluster together and mouse tissues cluster together, is in large part because most of the human tissues were sequenced in one lab, and most of the mouse tissues were sequenced in another lab. And so in some sense, the lab batch effect is almost completely confounded with the species effect that they were interested in. And so once you correct for example, lab-specific differences in gene expression, then you come to the opposite conclusion, which is that tissue-specific expression is more conserved than, or expression in the same tissue cross species is more similar than expression across different tissues within the same species. And so the point here is really that batch effects are huge factors of variation within many gene expression studies. And so you need to be very careful about batch effects when you design your experimental study. And so I just want to touch a bit more on
really obviously bad experimental design, because you can actually see a lot of experiments still being designed these ways. And so suppose, for example, that you wanted to sequence a bunch of samples corresponding to multiple conditions. So if you have two conditions, it would be like a case control experiment, or you could have more than two. So in this case, you have hypothetically 3 different conditions. And suppose that you weren't really thinking too deeply about your experimental design. And so what you did is that you sequenced your, for example, 3 groups of samples in 3 different batches, where you happen to group all of the samples from group 1 into one batch, all of the samples from group 2 into batch 2, and all of the samples from group 3 into batch 3. And so if you were to do this, what this means is that your batch is perfectly confounded with the group of samples that you're trying to sequence. And so any differences that you detect between, say, group 1 and group 2 could either be due to real biological differences between the 2 groups of samples, or they could be due to the fact that there's just differences between batch 1 and batch 2. And so, again, because essentially batch is confounded with group, then you can't tell what differences are due to batch or what differences are due to group. So any better experimental design, if you have 3 groups that you want to test and you want to do sequencing in, for example, 3 batches, what you could do is you could divide each group into two essentially replicates, where group 1 gets divided into replicate 1, replicate 2, group 2 gets divided into replicate 1, replicate 2, and group 3 gets divided into replicate 1 and replicate 2. And so then what you can do is you can basically divide the replicates among the batches such that each batch is composed of 2 different replicates together. And so basically you choose one replicate from group 1 to go with group 2, another replicate to go with group 1 and group 3, and then finally another replicate from group 2 and group 3 to form your 3 batches. And so 2 of the most basic kind of outcomes you can see when you, for example, visualize your samples after sequencing, is shown here on the right, is that in the case where there's no batch effect, then that means that your samples from the same group should essentially appear in the same region of your principal components plot. And so here on the PC plot on the right, you can see that all the triangles grouped together. So triangles refer to group 3. All the circles grouped together, and the circles refer to group 1. And all the squares are together, which represent group 2. And so this illustrates to you that there's basically no batch effect in this particular dataset.
And so just to wrap up the bulk RNA sequencing part of this lecture, just like with other types of genomic analyses, this question of which is more important, effect size or significance, also plays a role here. And so when we're talking about, for example, knocking down TFs and then measuring gene expression patterns before and after to identify targets of a TF, it's important to distinguish which genes are significantly differentially expressed but have small effect size versus large effect size. And so, what is more important here, the effect size or the significance? Another important point is that transcriptome profiling is in large part pretty useful, not just for doing things like differential expression testing between simple cases and controls, but also when you combine it with other technologies that we talked about in this class. So for example, you could combine RNA sequencing with not just knocking down a TF or knocking out a TF, but also turning on or off different enhancers to determine, for example, what is the effect of shutting down an enhancer or turning it on as a way of, for example, identifying target genes of an enhancer. And finally, as I pointed out on the last slide, in practical terms, a lot of the problems that people spend their time dealing with in RNA-seq analyses is really in trying to find ways to deal with batch effects. So now I want to spend just a minute to talk a little bit about non-coding RNAs. And so in this lecture, we're spending almost all of our time talking about sequencing RNA from protein coding genes. And so here I just want to point out that protein coding genes actually don't even make up the majority of genes in, for example, the human genome. And so here's basically a table of statistics from the GenCode database that basically maintains information about genes found in, for example, the human genome. And you can see that the protein coding genes actually only make up about a third of the known genes in the human genome. And so that means that there's a lot of genes and that are producing RNA that we're not potentially measuring when we're using when we're doing, for example, poly selection for RNA sequencing. And so this slide is basically here to illustrate to you that although again we focus primarily on mRNA sequencing in this lecture, there's actually a lot of different kinds of RNA molecules that the cell produces, and that people do actually measure and profile using RNA sequencing, but we just don't cover them in this course. So we're going to switch gears a little bit, and now we'll talk about single-cell versions of some of the bulk assays that we discussed earlier. In particular, we're going to go over single-cell RNA sequencing. And so the premise of the single-cell based assays are that tissues, for example, are highly heterogeneous. And so because bulk assays like bulk RNA sequencing require you to collect RNA from millions of cells, part of the problem is that if you're collecting measurements over millions of different cells, then essentially the measurement that you get at the end of the day is going to be an average, in some sense, over all of the cells that went into your assay or your experiment in the first place. And so when you're dealing with tissues where there's many different types of cells, then in some sense you don't really know exactly what you're sequencing because every sample is actually just a mixture of many different cell types, and the proportion of those cell types can change across your different samples. Right, and so as I mentioned, bulk assays collect measurements over millions of cells and average them together. And so the goal of single cell assays is that really you're trying to separate the contributions of each individual cell to your bulk sample. And so in the case of RNA sequencing, for example, suppose that you're interested in quantifying individual genes expression measurements. In a classic bulk assay, like bulk RNA sequencing, you would take your, say, for example, a primary glioblastoma, you would resect a small portion of that sample, and then you would collect all the RNA from those millions of cells and generate a single set of quantifications, or what's typically visualized as like a vector of measurements, where for each gene in the genome, you've measured how many times you've identified a read that's mapped to that particular gene. So in contrast, in a single cell-based assay, what you would do is instead of collecting all the RNA from those millions of cells and pooling them together, you'd, for example, basically dissociate all of the cells such that you can isolate individual cells, and you would punch those single cells through, for example, like a microfluidics device that can separate those individual cells and then basically tag all of the RNA from an individual cell with a unique barcode. And that would essentially allow you to separate out which reads came from which cells. And in that sense, then, instead of generating a single vector that quantifies each gene's expression measurement across the whole sample, you'd instead get like a table or a matrix where you could quantify each gene or protein in the genome for every cell that you sequenced in the assay. And so in that sense, you get around this problem of looking at heterogeneous samples, because now instead of averaging together cells from many different populations, you're actually able to sequence each individual cell from each cell population separately and basically unmix them. And so it's worth pointing out again that the idea that tissues and cells are highly heterogeneous really underpins a lot of the motivation of these single cell assays. And so if you think about cell identity and function, these are very kind of fluid concepts, and they in part are very context dependent. And so obviously, for example, it's important to describe and be able to characterize what general type of cell are contained within different samples. So in some sense, the function and identity of a cell is defined by whether or not they're, for example, like an excitatory neuron or an inhibitory neuron or some kind of fibroblast. But there are other factors which play a role in defining cell identity and function. So for example, if you're talking about pluripotent cells, or if you're talking about some kind of differentiation process, right? So differentiation is typically thought of as a continuous process where you might start with a kind of well-defined cell type, like a, say, induced pluripotent stem cell or an embryonic stem cell. And you might be interested in studying how that cell differentiates and changes identity as it differentiates from an iPSC cell to, for example, like an erythrocyte or a neutrophil. And so defining all of the states that a cell can exist between an iPSC state and, for example, an erythrocyte state can be really difficult because, again, that process of differentiation might take a cell through a possibly incredibly large number of distinct states, like hundreds or thousands of different states as it moves, through this differentiation process. And so, if you are trying to characterize what are the sequence of molecular events that lead a cell to differentiate from iPSC to erythrocyte, then you might have to, you could potentially be dealing with a problem where you need to be able to observe and sequence a cell in many, hundreds of different states along that path of differentiation before you can really understand how the process works. And so the point here is that to study that kind of fine detailed differences between individual cell states during processes that can be fast, like differentiation, doing this with bulk assays can be extremely hard, if not impossible. So over the past decade, a number of single-cell versions of the bulk assays that we talked about in this course have been developed. And so there are single-cell assays for doing DNA sequencing, ATAC-seq, bisulfite sequencing, as well as RNA-seq. And so of all of these different technologies that have been developed, the single-cell RNA-seq based assays are the most mature and definitely the most heavily used as of today anyways. And so for this lecture, we'll mainly be focusing on single-cell RNA-seq based assays. But again, it's worth noting that there's single-cell based versions of pretty much every major assay we talked about in this class. And so the figure in the bottom right-hand corner of this slide basically just illustrates to you how quickly this field has moved in the last 10 years or so since the single-cell based assays have begun to show up. And so you can see that one of the first whole genome based single-cell sequencing assays basically came out somewhere between 2009 and 2010. And back then they could really only sequence the whole transcriptome of 1 to 10 cells. Then nowadays you can see as recently as 2017, 2018, that in a single experiment, people can sequence now up to like hundreds of thousands or even a million cells. And so you can really see that these capture technologies have improved significantly over the years. Despite this kind of huge progress in the last 10 years of developing mature single-cell based assays, there's still a number of large challenges associated with single-cell based collection and analysis of the data. And so the three major challenges so far are basically, number one, challenges associated with cell isolation. And so it can actually be pretty tricky to dissociate bulk tissues into individual cells in a way that doesn't kill that many cells, but does successfully isolate individual cells. The second major challenge is basically due to the limited amount of material that you can get out of individual cells. And so one of the reasons why the bulk assays require millions of cells is because any given individual cell doesn't really contribute that much RNA or DNA or so on. And so even though some of the technological developments that have enabled single-cell assays to work basically are highly efficient at kind of not losing much RNA throughout the protocol, still there's still not a whole lot to work with when you're talking about material from an individual cell. And so that basically leads to implications in terms of downstream data analysis and how much effective data you can actually get out of individual cells. And so in terms of single-cell RNA-seq, there's a few different motivations for why people choose single-cell-based assays over bulk RNA assays. And so the first, as I alluded to earlier in this lecture, one of the major motivations is that biological samples in general are highly heterogeneous. And so because the bulk assays average molecular measurements over all of the cells that contribute RNA to the bulk RNA assay, then you kind of lose this distinction of all the different types of cells in your sample. And so in principle, with single cell-based assays, because you're sequencing each cell individually, and that means that you can kind of characterize what proportion of your sample came from different types of cells, and you can characterize the difference or the variation in gene expression that you might see between different cells in your sample. And so this also falls under this category of kind of deconvolving the heterogeneity of a sample is that these single cell-based assays are also increasingly useful for example, identifying rare cell types. So for example, if you're looking at tuber samples, basically in the field of cancer, some people think that some types of cancers are largely driven by a really small rare population called cancer stem cells. And so these cancer stem cells used to be impossible to detect in an unbiased way when you're looking at bulk tumors. But with single cell-based assays, it becomes much easier to identify a small population of cells present in your samples. And so the second major goal of single cell-based assays is really to perform what's called trajectory analysis. And so here again, the idea is that if you're looking at, if you want to observe continuous processes like differentiation, then with bulk samples, unless you do some kind of synchronization, it's basically impossible to really tease out all of the different molecular events that can happen along a continuous process like differentiation. This is in contrast to single cell-based assays, where, for example, you can take pluripotent stem cells, you can start the process of, you can initiate differentiation, and then you can basically just try to sequence tens of thousands of cells as they're differentiating. And basically when you then, the idea of trajectory analysis is that when you then kind of visualize the cells that you sequenced, because they all kind of are unsynchronized, in principle, if you sequence enough cells, there should be hopefully a representative set of cells that were in any given state along the differentiation process. And so in some sense, by kind of looking at a visualization of all the cells you sequenced, hopefully they can form some kind of branching pattern, or in what's oftentimes called a trajectory, which then kind of allow you to trace what happens as you move from one end of this trajectory or this differentiation process that you visualized to another. And so again, this is a process called trajectory inference. And so finally, people are also interested in really constructing and inferring gene networks in different types of cells. And so in the networks lecture, I briefly mentioned that one of the challenges of building co-expression networks is that you typically might need hundreds or thousands of samples in order to accurately build a network. And that, doing RNA-seq on hundreds or thousands of samples is very expensive. And so you can't really do that often. Single cell-based assays have kind of brought network inference back into style, in the sense that because you can now perform single cell assays on hundreds of thousands or millions of cells now, people have thought that, okay, well, maybe now I have enough samples to really accurately infer gene networks, whereas I didn't back in the days of just collecting bulk RNA sequencing data. And this is kind of true and kind of not true at the same time. So as you'll see in a few more slides, it is true that with single cell-based assays, you can profile hundreds of thousands or millions of cells.
On the other hand, you'll find that on average, the amount of information that you get on any given individual cell is not really comparable to what you would get from a bulk RNA-seq assay, just because there's such limited amount of material or RNA in any given cell. You end up getting less information per cell out of the single cell based assays compared to bulk RNA-seq. And so in the next few slides, we'll talk a little bit more about the different single cell capture technologies that have been developed. And so there's a countless number of single cell RNA-seq based assays that have been developed. Some commercialized, some not, some more or less at a higher throughput with less information per cell, and some that focus more on getting more information out of each cell, but at a lower throughput, so fewer cells per assay. We'll focus a little bit more on the main categorization of assays based on whether they do what's called 3' tagging or whether they profile full-length transcripts. So this slide gives a pretty incomplete now overview of different single-cell RNA-seq based technologies. And so the main distinction between the ones listed here anyways are whether or not the particular technology sequences full-length transcripts or only does 3' tagging. And so methods that assay full-length transcripts basically generate reads that can come from, in theory, any part of any transcript. And so they typically involve some kind of like fragmentation of full-length transcripts into different smaller fragments, which you then get sequenced, analogously to the bulk RNA-seq protocol we talked about earlier. And in contrast, the 3' tagging methods really only focus on sequencing a small short read out of the 3' end of transcripts. And so you can see in this table below that there's basically a kind of a inverse relationship between the amount of detail you get out of an individual transcript versus the throughput of the assay. So what I mean by that is if you look at the full-length based approaches, generally speaking, the throughput or the number of cells that you can sequence is typically somewhere on the order of at most like 100 or maybe say 1,000 cells per assay. Whereas when you look at the 3' end counting methods, they really allow you to sequence a few orders of magnitude more cells compared to the full-length approaches. And so, for example, split-seq, at least according to this table, allows you to sequence somewhere between like 1,000 and 10,000 cells. And that number is actually much higher nowadays. So you can actually use, for example, Chromium based approaches to sequence like hundreds of thousands of cells fairly easily. And so really, but if you look at the typical read depth per cell, the full-length approaches tend to give you like a higher number of reads per cell compared to the 3' end counting methods. And so really the choice of full-length versus 3' tagging methods is a choice of how much information you want per cell. So typically the full-length transcripts approaches allow you to get more reads per cell, and they also in some sense give you more information per cell or per transcript, because since your reads come from all over any given transcript in principle, that means that you can detect, for example, alternative splicing that occurs early on in the transcript. Whereas for the 3' tagging methods, because you're only sequencing a small portion of the transcript at the 3' end, that means that you don't see what happens in the 5' end. And so there actually are now protocols for doing 5' tagging as well, but they're not discussed here, and they would also suffer from the similar problem that if you only sequence one end, then you don't know what happens at the other end. And so another really important point I want to make here is that, again, the throughput that we're talking about here is on the order of like hundreds to like hundreds of thousands of cells. And so in comparison, some of the biggest gene expression based studies that used bulk RNA sequencing methods, such as the GTEC project that sequences different human tissues, even these kind of super large consortium that did bulk RNA-seq assays could only really sequence on the order of hundreds or now kind of low thousands of samples for the entire project. Whereas with the single cell based assays, you can really
sequence like 100,000 cells all in one experiment. And so the scale of the data that comes out of the single cell-based assays is just orders of magnitude more than bulk samples. So the DropSeq single-cell RNA platform, which was later commercialized by 10x into their Chromium product, is one of the most high-throughput, frequently used platforms for doing single-cell RNA-seq. And so we'll briefly discuss how it is used and how it works here. And so the idea is that you start out with a microfluidics device that basically has 3 input channels shown on the left. One that's used to pass in dissociated cells, another for microparticles and lysis buffers, and a third for oil. And so we'll discuss what the microparticles or beads are used for on the next slide. But basically, when you add the lysis buffer in with the oil in the cells at the right concentration and speed, this basically causes droplets to form, where on average, you hopefully get 1 bead in 1 cell within each droplet. Although sometimes you'll end up with empty droplets, or sometimes you'll end up with droplets with a bead and no cell. Or, for example, you'll get a droplet with a cell but no bead. And so the idea is that after the droplets are formed, the cells get lysed, and the mRNA contents get dumped into the droplet. And so the idea of the beads is that each one of these beads, or what are called microparticles here, have a bunch of primers hanging off of them. And each primer has a poly-T tail that basically allow them to get hybridized to the poly-A tails of mRNA from the lysed cells. And so the key idea here is that, and we'll show this later on the next slide, is that for every bead, all of the primers hanging off of that bead have a unique cell barcode, and it has the same cell barcode on each bead. And the idea is that this cell barcode basically allows you to later on identify that all the mRNA that hybridized to that bead came from the same cell. And so again, every primer hanging off of a bead has the same cell barcode. But on the other hand, as we'll again see later, every primer also has what's called a UMI, or unique molecular identifier, where hopefully every primer has its own unique UMI for a given bead. And so after the mRNA hybridizes to the poly-T primers on the beads, then the droplets get broken and the microparticles get collected and washed. And so here the important point is that even though the droplets are broken and therefore all of the different beads are pulled together, because each of the beads has its own unique cell barcode and all of the primers on that bead have the same cell barcode, then all of the mRNA that get hybridized to a single bead have to come from the same cell. And so the mRNAs then get reverse transcribed in bulk, and that basically forms what are called stems. And then a technique called template switching, which we won't go over here, is basically used to introduce a PCR adapter downstream of the synthesized cDNA. And so the cDNA get released from the beads, and then they get PCR amplified. And as we'll talk about on the next slide, the PCR amplification process is a bit more robust than the one typically used for bulk RNA sequencing. And because of the unique molecular identifiers, or UMIs, we can basically, in large part, address the PCR amplification bias that plagues a lot of bulk RNA sequencing assays. And so after we sequence the reads, then as usual, we can then map them to the genome or the transcriptome, and then identify which gene each cDNA came from. And so again, because each primer on the bead has the same cell barcode, that means for every cDNA that we sequence, we know which other cDNAs came from the same cell, and so we know their cell of origin. So the 10x Chromium approach is called the 3' tagging approach because these essentially full-length cDNAs are sequenced using, for example, paired-end short read sequencing. And so what happens is that on one end, you're going to sequence some barcodes that tell you the cell of origin. And on the other end, you're going to sequence part of the 3' end of the transcript. And so using that 3' end, that's how you're basically going to identify which gene the transcript came from. But what that means also is that because you only sequence the 3' end of the transcript, then
you may be able to identify the gene that the cDNA came from, but you won't be able to identify, for example, alternative splicing events that happened in the 5' end that are past the barcode that you sequence. So part B of this diagram shows you an actual illustration of the microfluidic device used in DropSeq. And so here you can see the beads basically come in through the central channel moving left to right. The cells come in from two channels, the top and the bottom, and oil basically comes in through the top. And basically the droplets form and move left to right as they get lysed, and then the mRNA gets hybridized, and so on and so on. And so part C of the diagram basically illustrates a single cDNA molecule constructed in this library. And so you can see that basically the reads labeled read 1 and read 2 represent a hypothetical paired-end sequencing of the single cDNA molecule. And you can see that basically the read corresponding to the 5' end of the cDNA captures both the cell barcode as well as the UMI. And the read coming from the 3' end of the transcript basically captures the 3' end of the transcript and is used to basically identify the gene that the transcript came from. And so part D illustrates a hypothetical library that corresponds to a whole bunch of cDNA sequenced from a given single-cell RNA-seq experiment. And so the important point to note here is that this is an example of the hypothetical library that you might generate, say for example, before PCR amplification. And so how we get around the PCR amplification bias is that when PCR is applied to increase the total number of copies of each cDNA, what you'll note is that every original cDNA molecule has a unique molecular identifier that tells you that corresponds to exactly one transcript in theory that was hybridized to one of the beads. And so even if certain cDNA molecules get amplified more often than others, you'll be able to tell what that amplification bias is, because if, say, the first cDNA molecule was amplified 10 times more frequently than the second one, you'll actually see the UMI barcode for the first cDNA molecule get sequenced 10 times more than the second one. But you can actually tell how many times each cDNA molecule was amplified, relatively speaking, because you'll just see the same UMI get duplicated over and over and over again. And so you can actually tell how many original transcript molecules there were, because that will correspond to the number of unique UMIs that you sequence in your library. And so again, after you align the cDNA to the genome or to a reference transcriptome, and you group them based on, you group cDNAs that have the same cell barcode together, because you know that they came from the same cell, and that allows you to essentially count how many times each gene is represented within each cell in your library. And that's how you get your table that represents your single-cell RNA-seq experiment that tells you how many times each gene was basically identified in each cell. And so the second metric that they also measured is what's called sensitivity, or what's precisely defined here as the molecular detection limit. And so the molecular detection limit is defined as the minimum number of input RNA molecules that you need in order to confidently detect a gene, or in this case, like a fake gene. And so higher sensitivity, or essentially a lower detection limit, means that you can detect very weakly expressed genes. And so you can see here that bulk RNA sequencing actually has relatively poor sensitivity, or correspondingly a high molecular detection limit. So that means that you need to sequence a lot of molecules, a lot of RNA, in order to detect weakly expressed genes. And so in contrast, most of the single-cell RNA-seq methods have a lower molecular detection limit. But again, you can see that even across the protocols, or even within a given protocol, you can see that there's a huge variation in terms of the molecular detection limit.
And so one of the most prominent features of single-cell RNA-seq data compared to bulk is the presence of zero inflation. So zero inflation refers to the fact that when you look at your data table that tells you how often each gene was detected in each cell, you can actually see a ton of zeros in this table. And so, for example, for data that comes out of like drop-seq experiments or 10x Chromium experiments, sometimes up to like 95% of your table are zeros. And so this is really striking. And so if, for example, you were to take two replicate cells and sequence them using, say, drop-seq, and you were to say, suppose you were to plot the expression level or the abundance of each gene across the two replicates, you basically get a plot similar to the one you see here on the right. And so in an ideal situation, this plot would actually be diagonal. So each point in this plot represents a single gene. And so ideally you'd see a line on the diagonal, which means that you get the same abundance measured in each of your two replicates. But in practice, you see this cloud, which means that there's a lot of kind of technical noise in these measurements. And furthermore, you can actually see two bands on the axes, so one on the x-axis and one on the y-axis. And what these bands represent is basically zero inflation or dropout events, where, for example, the band on the x-axis basically represents a whole bunch of genes where there was some non-zero expression measured in cell 2, but you measured zero for all of those genes in cell 1. And then similarly, the band on the y-axis represent genes where you had some non-zero expression in cell 1, but you measured exactly zero in cell 2. And so a lot of these dropout events are not that reproducible. So we'll talk a little bit about how these dropout events they tend to be correlated with poorly expressed genes. But in general, people think that one of the major sources of these dropout events is basically the reverse transcription step that happens inside the droplets. And so generally speaking, that reverse transcription step is thought to be very lossy and poorly efficient. And so basically a lot of poorly expressed genes essentially just basically get dropped out of the measurement in that case. And so again, the end result is that many genes have basically zero, they have zero expression in the table, whereas we think that they don't actually have zero expression. We think that they're being expressed at some level. So there's a number of sources of gene expression variation that drive the variation that you see between single cells. And so a large part about single cell data analysis is trying to figure out which factors are really driving variation in your data. And so in terms of technical sources of variation that you don't really care about oftentimes, or that you're not interested in, some of the common kinds of factors are things like batch effects. And so again, samples that are sequenced together in the same run tend to look more similar to each other than to samples in other batches. And so to mitigate this, people tend to try to multiplex multiple samples in the same run. So they sequence multiple samples in the same run so that you can compare samples within the same run without concern for that type of batch effect. In terms of library quality, we didn't really talk about how to dissociate tissues into individual cells. But this is a really tricky process because when you use too strong of a dissociation protocol, you can essentially kill a lot of cells and reduce your library quality. But on the other hand, if you don't use a strong enough dissociation protocol, that can leave you with so-called doublets, which are essentially pairs of cells that are stuck together, or triplets of cells stuck together, and so on and so on. And that essentially means that at the end of the day, if you're even able to push these doublets through your microfluidic device, your individual cell measurements are not actually of individual cells, but of clumps of them. So they're like mini bulk sequences. And so there's a lot of variations in the different types of library protocols that you can use. And you can choose among them depending on whether you're working with, for example, fresh tissue samples or fixed samples, or sometimes you actually want to sequence just RNA within nucleus as opposed to within an individual cell, and so on. And all of these choices basically affect your library quality. And so capture efficiency refers to how efficiently you can capture mRNAs once you lyse your cells. And so this can vary a lot depending on a number of factors, including, it turns out, how many RNAs are actually in the cells in the first place. And so it turns out, for example, that cells that have fewer total amount of RNA tend to have lower capture efficiency for some reason. And so in terms of amplification bias, I mentioned that UMIs are one technology that allow you to detect PCR duplicates and remove them after sequencing. And so in that sense, you can reduce PCR amplification bias, but you can't remove it completely, right? And so imagine, for example, that after PCR amplification, your library size is much larger than the number of reads you sequence. And so one problem that can arise is that, sure, if you sequence PCR duplicates, you can detect that and remove them. But the problem you have is that if there are certain cDNAs that are not amplified or amplified very poorly, and they're present in very small proportions of the total kind of population of the library, then you may just miss them in sequencing and therefore just not detect them at all. And so what's listed under allele intrinsic variation basically refers to this transcriptional bursting that I was talking about before. So again, there's a lot of stochasticity in gene expression that can now basically be observed in single-cell RNA-seq data. And so, for example, transcription for even constitutively expressed genes isn't really a constant process. There's oftentimes, for example, bursting of transcription followed by pauses of transcription. And similarly, processes like mRNA processing and maturation is a stochastic process, which in turn kind of gives a variation in what you measure with single-cell RNA-seq, because single-cell RNA-seq essentially measures like instantaneous measurements of gene expression patterns. And so finally, what's listed under allele extrinsic variation basically just refers to the fact that many people think that there's no such thing as a fixed cell type. And so cells are kind of, in their minds, cells are constantly kind of moving through some kind of continuous cell state space, and you can never really find two cells in the exact same state. And so furthermore, because things like cell identity and function are also functions of context and local position in the tissue, for example, since no two cells are exactly in the same environment, then that means that any two cells of the same so-called type are always kind of responding to different stimuli, and therefore they're also constantly varying. And so despite all of these kind of technical factors of variation and also the biological factors of variation that drive variation in expression across single-cell RNA-seq data, by and large, single-cell RNA-seq measurements agree with bulk RNA-seq measurements when your library quality is good and your number of reads is high. And so here's a plot where on the y-axis represents the expression level of a gene measured by bulk RNA-seq, and the x-axis represents expression measured through the Fluidigm C1 platform. And so each point represents a single gene. And so you can basically see that on average, there's a relatively high correlation between bulk RNA-seq and Fluidigm C1. You can see there's a bit more noise at the lower end of expression, so on the bottom left, whereas more highly expressed genes in the top right tend to be in more agreement between the two technologies. But on average, single-cell RNA-seq and bulk RNA-seq seem to correlate well when you have high read depth. And so here's a classic, what's called a t-SNE visualization of a single-cell RNA-seq dataset. And so in your typical visualization, you are visualizing a large dataset of single-cell RNA-seq, where the x and the y-axis are just kind of a 2D representation of your cells, and each point represents a single cell in your dataset. And in this particular case, a method called t-SNE is used to visualize your data. Other common software includes things like UMAP. These methods like t-SNE and UMAP tend to do a better job visualizing data compared to PCA because they're what's called nonlinear dimensionality reduction techniques. But they're less interpretable in the sense that we don't actually know what these individual axes mean. And so basically one of the points of visualizing your data are to basically see how cells cluster in your data set. So here again, I mentioned that each point represents an individual cell. And one of the first striking features about this visualization is that you can see that points seem to
points are not randomly distributed around this figure. Points are grouped into different clusters. And those clusters seem to correspond to the tissue or cell type of origin from which they were sequenced. And so basically visualization gives you a kind of first pass look in your data and allows you to kind of roughly guess how many kind of cell types are in your data set, where you might tentatively think that each grouping of cells might correspond to a putative cell type, because grouping of cells group together when they exhibit similar expression profiles. And so from this visualization is essentially like a starting point from which you can, for example, identify candidate cell types, and then identify, for example, cell markers or kind of signature genes of each cluster by taking each individual cluster and, for example, doing differential expression tests to try to figure out which set of genes are uniquely expressed in one cluster of cells compared to all other ones. And that in turn can then help you kind of try to identify what function that group of cells performs. Because if a particular cluster of cells only express, if they express one gene or set of genes uniquely and that are not expressed in any other clustering of cells, then that kind of tells you that that gene or set of genes are probably a unique function, represent a unique function of that cell or that group of cells. And so kind of through comparing different clusters of cells and trying to figure out which clusters express which cells, or which clusters express which genes, then you can kind of guess to see what kinds of cells you've sequenced in your data set. And so again, trajectory inference is really about studying how cells decide cell fate. Right, and so here, the diagram on the right represents single cell data collected from cells that are differentiating from a pluripotent cell towards a myoblast. And basically you can see that with a high enough number of cells sequenced, you can see that when you visualize the cells in a 2D scatter plot, the cells kind of form a nonlinear line that starts at the red points and ends at the blue points. And so trajectory inference refers to a bunch of computational methods which are oftentimes applied to basically look at these 2D scatter plots and basically draw these so-called trajectories or differentiation pathways, where the start of the trajectory, or what I've labeled here is time equals 0 for pseudotime, represents the start of, for example, differentiation, and the end of pseudotime, or where time equals n, is basically where the computational method thinks that differentiation basically has stopped. And so trajectory inference just refers to the huge number of methods available for finding these kind of trajectories and doing some downstream analysis to figure out what kinds of genes are changing in expression as you move from the start of differentiation to the termination of differentiation. And so again, something I want to mention is that we talked about single-cell RNA-seq in this lecture and mainly focused on that. But again, there's pretty much single-cell based assays that are being developed for most of the assays that we've talked about in this course. And so again, there's kind of corresponding methods for doing methylation profiling at the single-cell level, ATAC-seq, ChIP-seq, Hi-C, DAM-ID, and so on and so on. And so it's, there's just, this field is just moving at an incredible pace. And so just like for single-cell RNA-seq, a lot of the methods like single-cell ATAC-seq, Hi-C, and ChIP-seq, they suffer from a lot of these problems related to low input material. And so that's why these methodologies are currently still immature, in the sense that while you can technically perform the assays at single-cell level, whether or not you can extract the same information out of those single-cell assays is a whole other issue. And so in terms of summarizing the single-cell RNA-seq portion of this lecture, basically the point that I've tried to make is that single-cell biology and related assays reveal a lot of information that you can't detect from bulk data. Some of the main current applications of single-cell RNA-seq are to study heterogeneity of bulk samples, look at the dynamics of transcription and gene expression and identify new cells or do trajectory inference. From a practical standpoint, the data that you get from single-cell experiments are still very much more noisy than compared to bulk assays, and they're much more difficult to analyze. And so the so-called software protocols for doing a lot of these downstream data analysis tasks are still kind of being worked out in the field. And a lot of the methods are still underdeveloped. Some things that I want to note that we specifically didn't cover in this lecture are, number one, the ever-growing set of single-cell based protocols that measure spatial transcriptomes. And so I said that for a lot of these single-cell based assays that we talked about, one of the first steps that you have to do is you have to dissociate cells from their complex tissues into single-cell suspensions. And one of the problems with that is that you lose the original spatial positioning of the cells in, for example, tissues. And so for a lot of tissues, for example, like brain or liver, a lot of the functionality of a cell depends on its context, and so it depends a lot on what other cells are around it, and so on and so on. And so there's a lot of technologies now being developed where you can actually both do single-cell sequencing, RNA sequencing, or at least you can profile the expression level of a small number of genes and actually maintain their original position in the tissue. And so in that way, you can get both the expression profile and the position of cells in tissues, and so you get a lot more information out of that. We also didn't really discuss multimodal assays, and so multimodal assays refer to single-cell sequencing protocols where you collect more than one type of data. And so, for example, scRNA-seq refers to technologies that allow you to both measure RNA sequencing and certain protein measurements of the same cell. There's assays that allow you to do both ATAC-seq and RNA-seq at the same time for individual cells. Assays like patch-seq allow you to simultaneously measure RNA-seq of a neuron, measure electrophysiological recordings on those neurons, and measure the physical morphology of those neurons all at the same time. And so there's kind of growing interest in these multimodal assays because they allow you to connect both kind of like RNA and like ATAC-seq or cellular phenotypes on the same cells, and therefore kind of build models about how variation in different molecular phenotypes covary. We also didn't talk about a lot of the even the kind of more mechanical details of single-cell RNA-seq. So, for example, how differential gene expression tests kind of vary for single-cell RNA-seq data compared to bulk, or even how quantification is dealt with. So even coming up with metrics for how to quantify gene expression is different for single-cell RNA-seq compared to bulk RNA-seq, for example. But we didn't really touch on that in this lecture.